right, I guess we can start now since there's a pretty good amount of people. So, hello everyone, welcome to the Breakout. This is with Professor Evelyn Yim. Professor Evelyn Yim is an associate professor at the University of Waterloo in the Department of Chemical Engineering. She did her PhD in biomedical engineering at Johns Hopkins University before doing her postdoctoral training, also at the Johns Hopkins University. She's also worked in Singapore in NUS, National University of Singapore, and done a lot of research in many different research institutes. So today, Dr. Yim will be talking about nanotopographical regulations on cell behaviors for tissues engineering applications, and this will be a very exciting talk. So I'll hand it over to Professor Yim to start.
Okay, thank you very much, Ryan. And I would like to thank the organizers who invite me to this very exciting event. And I would like to also thank every one of you who is joining this session, staying behind in this last breakout session. So as Ryan introduced that, I will be sharing with you about our work on nanotopography, on how we apply it to tissue engineering application. So stay tuned if you don't know what is nanotopography. You may have already have an idea what is tissue engineering and regenerative medicine, especially after the exciting panel discussion section. But I realized that I'm not sure if every one of you knows the concept of tissue engineering. So let me quickly walk through it. So the tissue engineering principle is proposed by Dr. Langer, who you have heard his keynote, as well as Dr. Vicanti. Back in the 90s, the concept involved isolating cells from a patient or a donor, and then we will use protein, other signals, and biomaterial to build a functional tissue. And using this functional tissue, we will use these functional tissues to replace or repair any tissues that is diseased or damaged. And other strategies that has eventually evolved is we also isolate cells from a patient or a donor, and then we will manipulate the cell function, and we will implant these functional cells back to the patients. So sometimes we refer it as cell therapy, and collectively we call it the regenerative medicine. So as the panel discussion has also mentioned that there are some key components for tissue engineering and regenerative medicine. One of the area that we are interested is the cell interaction with the extracellular environment. We know from Dr. Langer's section and also from media that drugs like chemicals like drugs, even the mRNA vaccine, proteins, they could be used to regulate cells' functions and behavior. Some of the extracellular matrix or the cell local environment signals, such as the rigidity or the mechanical property of the material or the microenvironment, could also regulate cell behavior. Our lab is particularly interested in one property of the extracellular matrix environment or the local environment of the cell is called the topography. And we have studied how this topography of the local environment will regulate stem cell differentiation, cell proliferation, cell function, or even disease development. And we are particularly interested also in nanotechnology, because nanotechnology is the components allow us to fabricate different type of topography, not only in a one-dimensional, but we can have topography patterns on a non-planar substrate. We could also have multi-layer or a more sophisticated architecture like a hierarchical structure, or we can even have detectable structure. Why are we interested in topography, and what is topography? Topography is a fancy word, but it is referring to the surface structure. So shown here is the electron micrograph of the extracellular matrix or extracellular environment. These two are the extracellular environment of a blood vessel, of a capillary in the kidney. And then you can appreciate the rich topography of the structure in the environment. Another well-known example is the cornea basement membrane, one of the cornea structure. You can also appreciate the richness of the nanostructure in the cell local environment. So okay, after we identify this topography, what we can use for, because we have identified that this type of topography, if we replicate it on synthetic or natural material, we can use it to control cell behavior. Our lab has been focusing on three areas of application, which I aim to, and I hope to give you an introduction on all three very briefly today. One is the neuronal differentiation of adult and pluripotent stem cells. The second is the application of the topography on developing material for vascular tissue engineering or vascular graft development. And the last application I would like to share today is how to apply this material topography or material nanostructure for cornea regeneration. So before I start going through the work, I would like to thank my team member, collaborators, as well as the funding, because I may run out of time at the end. So for the neuronal differentiation and mechanical biology work, I will be mainly showing the work from Solida, Kenneth, and Ben. And then for the cornea project, I will be mainly showing the work from Rizwan. And then for the vascular work, we'll be mainly sharing the work from Marie, Grace, and Yuan. And of course, this work we will not be able to do all this work without our amazing collaborators as well as our funding sources. So first, let me start with neuronal differentiation. We first studied about the effect of topography using neuronal differentiation as a model. This particular work, in this particular I show here, we studied the differentiation, the neuronal differentiation of human embryonic stem cells. That neuronal embryonic stem cell and pluripotent stem cell has the abilities to differentiate into almost any type of cells in our human body. But guiding stems to the forming neurons is actually particularly challenging. So by studying them on different type of topography, and we have developed a microarray that allow us to have a high throughput analysis of the cell behavior on a different type of topography at a more efficient manner. So my student Solida has studied the human embryonic stem cell on this type of microarray, we call it a mark chip, and we find that pillar structure are particularly favorable for astrocyte differentiation, versus line structure, or we call it the grating structure, favors neuronal differentiation. It also helps to enhance neuron maturation, which means that the period of differentiation becomes much shorter and we can generate neurons in a much more efficient manner. We've further applied this principle in a more recent work where Kenneth, my postdoctoral fellow, has applied these platforms to directly differentiate human-induced pluripotent stem cells to form dopaminergic neurons. Dopaminergic neurons is an important type of neurons involved in Parkinson's disease. In this case, we actually separate the procedure into two stages. The first stage is the lineage commitment stage, and the second stage is the maturation stage. We also find that at different stage, different topography signals will be needed to induce the cells to function. So in short, at the end of the study, we find that indeed topography, in this case grating structure, could significantly enhance the percentage of cells showing tyrosine hydrolase, which is a marker for dopaminergic neurons. Not only we have more dopaminergic neurons, when we look into the electrical physiology characterizations, we found that there are more neurons capable to fire repetitive firing action potential, which is a functional indicator that these cells are more active, more functional, having a more mature electrophysiology. So this is a more useful neurons. So all this has suggests that topography could enhance neuronal differentiation, and choosing the correct topography, we could enhance the efficiency as well as the percentage of generating neurons. Okay, next, let me change gears to talk about the cornea. Just as a brief introduction, cornea is the outermost layer of our eye, is avascular, and the transparency is one of the characteristics to allow us to see, to maintain the vision. Although the cornea is transparent, it is actually a highly cellularized structure. It has 3 cellular layers. The epithelium is the outside or anterior layer. The stroma is the thickest middle layer, and endothelium is the posterior or the inner layer. One of the applications our lab has been particularly interested is to reconstruct the corneal endothelium. So corneal endothelium, again, is the posterior layer that helps to provide nutrients to the cornea, as well as to maintain the cornea transparency. However, one characteristic of cornea endothelial cells in humans is that they lack the capacity to proliferate in vivo or in vitro. That means our cornea endothelial cells in our eye, they actually will not have the capacities to grow in terms of number. So as we age, we can only lose them, but we cannot regenerate them. And unfortunately, cornea endothelial dysfunction has been one of the leading causes of corneal blindness. And the main treatment will be cornea transplantation. So one of the early motivation of our work is to see if we can develop platforms to allow us to proliferate these primary human corneal endothelial cells in the lab in vitro, so we can generate more functional cells for cell therapy. So in this study by my former student Rizwan, we have studied the primary corneal endothelial cells on unpatterned tissue culture plastic with different type of pattern like nanopillars, micropillars, microwells, and we find one combination of micropillars could significantly enhance the functional markers, the tight junction formation in the endothelial monolayer. What more exciting is that we also find that this micropillar structure could significantly enhance the proliferation as well. But what happens if we want to form or tissue engineer a corneal endothelium and transplant these corneal endotheliums to the patient? We will need a transplantable polymer to function as a cell carrier. So again, Rizwan has further developed this platform to micro-patent these nanopillars into a gelatin methacrylate, or we call it a gel film. And this picture shows that we can implant the gel film at the back of the cornea. And not only the nanopatents on the gel films can also trigger and enhance functions of the corneal endothelial cells. In a 4 months implantation in the rabbit cornea, we do not observe any inflammation, and the cornea still remains transparent after the 4 months implantation. So lastly, in the last 5 minutes, I want to wrap up in the last application, which is our work on developing small diameter vascular graft. In many different vascular disease, which cardiovascular disease is one of the leading cause of death around the world, unfortunately. In different type of vascular disease, small diameter vascular graft is needed. For example, in peripheral arterial disease or peripheral vascular disease, which is one of the leading cause of limb amputation in the Western world. And other disease that also need small diameter vascular graft will be coronary and carotid arterial occlusion. So synthetic graft, like polymer synthetic graft, has already been used in the clinic, especially the one that has large diameter. So when we talk about small and large here, large diameter refers to the internal diameter larger than 6 mm. And when we are talking about small diameter, we are referring to the diameter, the inner diameter smaller than 6 mm. Synthetic vascular graft has also been used for small diameter application, but their failure rate is very high, mainly because they have short-term thrombosis problem. There's an increased rate of infection. People also observe a chronic inflammation response due to the graft material. And one of the speculation also is those polymer like EPTFE and DAC1. There's a significant compliance mismatch to the native vessel. So to address some of the problem, we started a collaboration with our collaborator, Dr. Catherine Lavisage at Instrom in France, to develop a vascular graft material with polyvinyl alcohol. The system used polyvinyl alcohol cross-linked by sodium trimetaphosphate. And the advantage over here is that we can allow us to tune the mechanical property to match the compliance and the mechanical property to the native vessel. So in our early work, we have done implantation of our vascular graft to the rapid femoral artery, which is smaller than 1 mm in diameter. So what is interesting here is actually very encouraging. So after 2 weeks of implantations, we are using digital subtractions angiography at 2 weeks. We actually observed the graft to remain patent. That means it remains open to allow blood flow, which is very good. However, we observed one problem is that because PV is very hydrophilic, it has a very hydrophilic surface, we couldn't observe the formation of an endothelial layer in the lumen, which has been shown to be critical for long-term stability and long-term success of vascular graft. So some of the proposed solutions we have will be whether we can do surface modification or if we can apply micro and nano patternings through the lumen. So indeed, we have developed a method to apply micro and nano patterns to polyvinyl alcohol. And we can also apply the pattern in the lumen of the very small diameter vascular graft. So here is a 2 mm internal diameter vascular graft. And then we use a rat model to implant this luminal patterned 2 mm diameter vascular graft in the aorta of the rat. And after 20 days, without any anticoagulant and antiplatelet drug, this graft will still remain open. And what is also encouraging is we see signs of endothelial cell, which is detected by immunofluorescence staining after the 20 days. But however, we still haven't found a perfect solution. Although we find some endothelial cell form, because PV is still too hydrophilic, even the topography or the nano patterns can help. We want to have the surface fully covered with endothelial cells. So my PhD student here in Waterloo, Yuan, she has further modified the PVA surface with a polysaccharide called fucoidin. And from this picture, you can see that the modified surface could enhance the cell adhesion much more significantly compared to the surface that is not modified. We go ahead to also compare the blood compatibility. We call it the hemocompatibility. One problem of enhancing surface for cell adhesion is they often will also activate platelet activation or blood coagulation. But in this case, we find that the modified PVA is comparable to our clinical control, which is the EPTFE. And in an in vivo study, we find that the modified PVA is the patency rate or the rate of the vessel remaining open at the end of the 4 weeks is as good as the clinical control, which is the EPTFE. So it's almost time's up. So to conclude, I hope that I have convinced you that cells material interactions can be engineered. So we could engineer how the cells respond to the biomaterial by modifying the material we want to use with nano patterning or nanotopography, plus the chemical modifications to enhance different tissue engineering application. I showed you very briefly the example of how we apply the nanotopography to enhance neuronal differentiation, how we applied it in small diameter vascular graft, and how we apply it in corneal endothelial regeneration. What I didn't show you, and you are welcome to ask, is you may want to ask how does it work. We also have done the study to understand how it works, and we show that the topography signals from the materials will trigger cells respond through force-mediated pathway, meaning that they will change the force transmission in the cell cytoskeleton through focal adhesion pathway. So with that, I would like to thank my students and collaborator, and I would like to thank you again for your attendance. Just a brief advertisement is that if you're interested in this session, you may also be interested in the Canadian Biomaterial Society conference that will be held next year. Originally, it will be in person in University of Waterloo, but now it will be online. And if you're a student from the University of Waterloo, you let me know if you may be interested to volunteer. We will look forward to participate. And although I have prepared some slides to talk about how I become a biomedical engineer, I think I should open the floor for question. Okay. I need to see the screen first.
Yeah. If you guys have any questions, feel free to type them in the chat and I'll read them out.
Oh, hello, Professor Sefton.
Yep.
I guess while we are waiting for questions, or if there's any questions, I can also talk about how I become a biomedical researcher. I did my undergrad study in University of Toronto, and at that time we didn't have biomedical engineering program, so I did my undergrad in engineering science, and then I did my master with Professor Sefton, and then I did my PhD in Johns Hopkins.
Yeah. We have a question. So the question is there any specific pattern that works for all types of tissues?
Good question. Short answer is no. As I mentioned, I hope I showed in the example is that the patterns are tools that trigger a particular cell response. So you can imagine that it's like different apps for different functions. So you have different patterns for different function. Some of the patterns has been known to trigger uniform, not uniform functions that commonly observed in different cell types. For example, for the line patterns that I showed, they will trigger elongation of the cells in most cell type. But whether this is what you want is a different question, because in some other applications, this may not be what you want.
Oh, we have another question. How can we resubdivide the decellularized organ, which consists of various tissue types?
That's a very interesting question. The decellularized organ, I suppose you Holder, you will know how decellularized tissue and organ is being fabricated or made by lysing the cells or removing the cells from the tissue and organ. So one thing that is important is this tissue and organ should have an intact vasculature that is preserved. And then using these, I think most of the current strategies is to use the existing vasculature system that is preserved in the decellularized tissue and use these vasculatures to for the cell seeding. Can the surface of the patterns be functionalized for better outcome? Yes. So I hope the last, I know it's very long presentation, but I still want to put it in the last presentation. The last point is model for the PVA application. We like the mechanical property, but the material may not be very good for blood connecting applications, although the topography helps. But then we find that it's still not the most ideal for cell interaction. So even with the patterns, we have to chemically functionalize it. And in that example, I shows that we functionalize it with a polysaccharide called fucoidan. Is it possible to very briefly mention one or two highlights? Okay. The first key point is then what I mean nanotopography is the surface structure or the architecture, the 2D to 3D nano architecture in a cellular local environment. If we understand how cells interact with this local environment, and how these local environments could trigger signals in the cells, we can use and apply this type of architectures to biomaterials to trigger the same response. And the second part is similar to a previous question. We will find, depending on what our application will be, whether it's tissue engineering, drug delivery, or depending on which tissue, we will find the ideal structure for the particular application.
Yeah, all right. Thank you, everyone. And I think that concludes Professor Evelyn Yin's talk today. So thank you so much, Professor Yin, for coming.
Thank you very much, Ryan.
Thank you.
Thank you.
Yeah, so everyone, if you guys want to head back to the main stage, the closing remarks are about to start.
Okay, thank you, everyone. Bye-bye. Have a good weekend.